Welcome everyone to the November 2023 cross-disciplinary seminar series on metabolites. This is hosted by the Office of Integrative Medicine and the Department of Biochemistry and Molecular Medicine. It's my pleasure to introduce Ali Ranavard, an assistant professor of the Biostatistics and Bioinformatics Department at the Milken Institute School of Public Health. Ali received his PhD from New Mexico State University in computer sciences, applied statistics, and bioinformatics before moving to Boston for his postdoc. There, he was at the Broad Institute and Harvard T.H. School of Public Health, where he was performing multi-omic data integration to investigate the role of metabolites and microbes in inflammatory bowel diseases. And he also worked on computational metabolomics data processing in microbial strains using shotgun sequencing techniques. In 2019, came to GW, and he's been developing really innovative computational machine learning, deep learning statistical methods for the purpose of achieving actionable research outcomes in health and disease using these high dimensional omics data sets for input. So in particular, his lab's really been focusing on single cells, how microbes and metabolites can be used to investigate problems, really thinking about them as nice representative snapshots of human health and disease. So his lab's using a lot of these computational tools that he's been developing to characterize the behavior of the metabolites and microbes in various disease settings. It's been very productive in publishing projects that he's been driving and leading, as well as numerous collaborative efforts while he's here at GW. He's already secured a lot of funding from various different sources, including NIH, NSF, the Gates Foundation, and he's built a really vibrant research program that in and of itself feels like it's the size of a small department. He has very, very many trainees that he works tirelessly to make sure they're being mentored well. He's very collegial. He's established these collaborations, as I've stated, including working with my team on a project. So today, Ali's going to be speaking to us about computational approaches he develops and how they're able to be utilized to infer biological insights from metabolomic data. So Ali, thank you very much for joining us today. We look forward to hearing about your work.
Excellent. Thank you so much, Brett, for the kind introduction and excellent job of doing that. You know, I should have recording you for my summary. So as Brett mentioned, my group we are in the Milken Institute School of Public Health in GW. Our department is about 4 years old now, Department of Biostatistics and Bioinformatics. We are part of the Computational Biology Institute. It's a research institute that we have a lot of collaboration within and outside of George Washington University. And through these collaborators, we have done several works. Today, the focus of this presentation is on techniques, computational techniques we have developed, that they are generic techniques, but the focus of my group is on metabolomics. So we have applied and used them ourselves in our projects and collaborators as well on particularly metabolomics data. So our goal in general is to understand the metabolomics role in health and how we can use them to find the patterns and answer biological questions. Today, the focus is introducing few of those computational techniques. Keep in mind that our group, we do a lot of investigation in health, but that's not the focus here. The focus is introducing the software and the tools that we have developed, and hopefully you find them useful for your research or the collaboration you have, and we'll be happy to support you. If you find them interesting at the end of this talk, you can use them. So I'll be skipping a little bit because of the time, but hopefully I'll cover the main point of this presentation. So just to give you why we are interested in metabolomics in general, here I'm showing the research community using the progress in terms of using omics technologies in general, how they have been progressing over the years. You see in the x-axis we have years, and the y-axis is the number of publications. We use the PubMed database and we visualize them. So each subplot shows a different omics, and each color in the bar plot shows the amount of or the number of publications that they used specific technologies under those omics categories. For example, metabolomics, that's one of our lab focus. We use the LCMS, liquid chromatography, mass spectrometry techniques that we have seen has increased over years in terms of being used to investigate human health. So that's the one thing that we are focusing on metabolomics. Also, the metabolomics specifically, if you look at them based on the technology they have been used to investigate different human health conditions, we see that has been increased. For example, COVID-19, we know that is a new disease, 2020 and over. So we see that metabolomics has been used to investigate this disease. The last bar is shorter compared to the others. That's because we made this figure early on in this year. So you see the metabolomics has an excellent interest in our community, research community, to investigate human health. And we want to introduce few specific software or computational techniques that they're based on background is machine learning and statistical approaches to investigate specific patterns in the using the metabolomics data. And you see here LCMS is one of the popular metabolomics profiling techniques that is the most of focus in our group as well. So when we talk about the metabolomics, we can consider them as, or they are omics data, and they have some properties. These properties are very important for us, and we need to make sure that we consider these properties in our data. They are high dimensional, they are zero inflated, and each of those needs specific curation techniques. For example, high dimensionality is when you have fewer samples than the number of features that we measure. So today I'm going to talk about 3 techniques if we get the time. If not, I'll briefly explain them and I pass it, then we complete the whole introduction, and in the future we can extend them if you need them, and we can support you and meet you to use this software. So I'm going to mostly skip the application part because of the time. I'll focus on the first part of this presentation, which I'm going to introduce 3 softwares. One of them is Tweedieverse for differential analysis. Is a very specific task. We do it is very generic for most of the study. We apply that. I'm going to introduce Tweedieverse. Then I'm going to talk about the massSight, which is allow us to combine the metabolomics data, and I'll talk about why that is challenging and why massSight is very powerful. And waveome, which is another software when you have a longitudinal metabolomics or any omics. Remember, the Tweedieverse and waveome are generic. You can use them for metabolomics or other omics as well. massSight is specific for metabolomics that has been processed using the LCMS to combine them. So differential analysis is a common task. The goal of differential analysis is to find specific omics features that they are significantly changed during the health conditions. That is a very common task, but it has some statistical and computational limits in omics data when we apply these existing techniques on the omics data. One of them is these data are high dimensional. One of them is they are zero inflated. And one more important thing in the omics data is they break assumption about the type of test we do. Most of the statistical tests we do, they have some assumption about the data that you analyze them. But omics data, because they are high dimensional and there are many of them, they don't follow the same distribution, statistical distribution to know which statistical test is appropriate. So we introduced Tweedieverse, which try to tackle those challenges. So there are 3 main concepts or challenges when we try to analyze the omics data. One is we need to normalize them. We need to pick how we should normalize the data. One is what statistical model is important to use, and then also we want to have a good power, meaning that we want to find as much as we can about the statistically significant association, and in the same time we want to control the false discovery rate. So these are many challenges with omics data that I mentioned here, and we are trying to address those using the multi-omic, using the differential analysis technique, Tweedieverse, that we have done. So here I'll show improvement in these techniques over many years, and they are lacking, there are several or challenges or lack of gold standard method, which method I should use, what is the standard that I should use for the omics data. And there are many issues with benchmarking, how they perform in different conditions. So Tweedieverse tries to tackle these questions, and the way it does it, it uses the Tweedie model. And the Tweedie model, the way it works is, and this is the take-home message from the Tweedieverse, which we initially thought to apply it on the microbiome, then we tested and applied on single cell, and now we are using it for metabolomics, and for all of those works very well. And the issue that initially I mentioned with the omics is that they follow different distributions. And Tweedieverse, the way it works, it tries to find the distribution or characterize the distribution of each feature independently, and then based on their distribution, they apply a statistical test and finds the differential analysis. That's the unique thing about the Tweedieverse, which uses the Tweedie models, has three parameters, mean, variance, and the Tweedie index, which they characterize the distribution of each omics feature that we test. And based on that, they find the differential analysis that we can use for association among all the omics. So today I'm going to just show a very quick application to show how it works and how we have used it. We used the Tweedieverse for many different applications. Here is one of them, which is a new project in our group with the Children's Hospital, that we are investigating the MEKi, or MEK inhibitor for the NF1 Neurofibromatosis type 1, which is a disease that causes is a type of cancer in the pediatric or in children's, and then it causes by the mutation in a specific gene, NF1, which we're going to look at, and causes the cancer mostly in the skin, like lump on the skin. You see several issues of that. And this gene is in the chromosome 17 in a specific location. And when it gets, we see the mutation, we see this NF1 disease. And the way this disease works is when the NF1 gene gets lost in the process of the metabolomic process, it causes increase in MEK, which is a protein in this pathways that we are showing. So when NF1 is lost, then MEKi increases. Now there is a treatment for controlling the MEK, which is the MEKi, and recently approved in 2020 by FDA. Our goal in this study was to look what are the changes that this MEKi causes after the treatment, and how we can use and understand the mechanism behind this treatment. So we want to investigate and follow up on this treatment. And this is a study, a little bit complex study to investigate the metabolomics. So basically we had the human subjects and we had the animal subjects, and we tried to have several controls because metabolomics are very sensitive in terms of issues that when you process them. So they are very sensitive to the technology and the people who have processed the data. Many batch effects could cause changes in the metabolomics data. So we try to look at different, here in this study, look at the vehicle effect on the metabolic controls when we the subjects, they don't have the disease health conditions, and we treat them with the MEKi, how those effects. And we have a group of graduate students with the help of Miriam Bornhorst from the Children's. We try to investigate this. And then you see the setup is longitudinal. In the same time, we have a different controls, and each of them has a different purpose in terms of the question we want to ask. Just briefly here, when I look at this metabolite in this MEKi, this is the key for the Tweedieverse tools. So the Tweedie index that I'm showing here, so this is a work with me and Himmel, both we developed the Tweedieverse. So when we look at the individual features in this study, this histogram that you see shows that the features in the study, they follow different distribution. The Tweedie index that you see in this plot reflects the type of distribution they have this omics feature. And we see there are many different distribution, and if there's one Tweedie index, this is a Pausen distribution. If it's two, is a gamma distribution, and we see many of the metabolites, they follow the gamma distribution, and that causes how we should analyze them. Here we look at the immediate effect of MEKi based on the specific groups we have in the study, and we saw that there are many metabolites that has been affected after treatment using MEKi in mice. So we apply that, and here what you are looking at different effect size or changes in different metabolites when we compare the MEKi versus vehicle. And you see we have labeled them to show the size and the significance of these effects. And you see many metabolites that we expected or they were new, novel. You could find them using Tweedieverse, their change, because again, this high dimensional data, Tweedieverse, in this high dimensional data, Tweedieverse allow us to apply this technique and capture these specific changes that sometimes they are get lost if you use a traditional methods that they follow one assumption about all the features. Okay, again, I'm gonna escape this a little bit. Just I'm highlighting a few metabolites that they have an important effect in the disease. Some of them are well known, like this serotonin is a well-known neurotransmitter that we see here. It changes in MEKi, increases after treatment in MEKi, but it gets lost when you use a vehicle. So that was interesting. There are many other metabolites that we are investigating that they had a specific meaning to us, and we are trying to understand their function during the NF1. Here you see a bigger setup. Here we are showing the metabolites effects on the MEKi for two different for a specific question. One question we had here about the MEKi treatment was to see if the metabolites that they change after the treatment using the MEKi are reversible or not. Do they get back their change to original situation they had? And we see, yes, the effect of MEKi are reversible. They can change over time. And that's why we had the longitudinal study here. We see the effects, but this effect can be lost over time if you don't continue the treatment. So those are a few examples here. We look at those changes in the human. Remember, we had the animal model and human, and then we see some of them, especially the metabolites in human. Also, they point to lipids that we show. Many of the metabolites that we saw change on them, they were lipids, and we see the lipid metabolism also follow the same pattern that we saw in the animal model. So just to give you a quick usability of this Tweedieverse. So we have applied on many studies, and I'm just here highlighting it on COVID-19. Our goal was to find the biomarkers at the time to see if they are metabolites that they can follow that they can be indicators for testing and diagnosing COVID-19. So the previous one was to characterizing the function of MEKi. Here, the goal of using Tweedieverse to find the significant metabolite changes in severe, non-severe, and non-COVID versus healthy, to find metabolites that they can be used as an assay for diagnosing COVID-19. And here we see that there are metabolites like cytosine that is being well established now, that changes in the metabolite in the COVID-19 patients, regardless of their severity. And the patients who they have a similar symptom, we call them non-COVID-19, but their test was negative, which they didn't have the COVID-19. We don't see that change for them. These are perfect examples that they can be used for diagnosis because they are indicator for COVID-19, but we don't see the change in the people with the similar symptom and not COVID. So we have shown the applicability of these tools in many different cases and applications, and here I showed two of them. And there are many other things. So the goal here was to test the association between these omics and find the effect size and the changes in different health conditions. And here this plot shows that the assumption we have that all the metabolites, all the omics features, they follow the same distribution is not a correct assumption. You see here in infant stool, microbiome or breast milk microbiome or metabolites, or in COVID-19, you see the distribution of the Tweedie index is different for different omics data. So that ensures that we need tools like Tweedieverse that look for the distribution of each omics feature individually and test feature base. Basically, for each feature, we need to first understand the distribution using the Tweedie models, then find their effects to have a correct statistical test for them. So that was one of the tools, Tweedieverse, to do the analysis of the differential, to do the differential analysis. So, and that's a common task for many studies, and Tweedieverse is a R package, can be used and installed to analyze the data, and we have tried to put a lot of tutorial for that, and is available in a GitHub with many examples. Now I'm going to shift to another tools named massSight, and the motivation behind massSight is this. Because there are many metabolite based studies, and we look for the biomarkers using the metabolites for different health conditions. If we want to save energy and expenses, or in general have a multi-evidence for a specific biomarker, it's important to do the meta-analysis. To do the meta-analysis, most of the time you need to combine the data sets that they come from different studies. So, but that's a very difficult task in metabolomics data. In the microbiome data is simple. You combine using R or using any other tools to combine directly the profiles you get from the data set. But in the metabolomics, that's not the case, because we don't know which feature map to which feature in the second data set. If you, let's say you have two data sets, one from study 1, one from study 2. Many times these metabolites, they don't have name even. And if you want to combine them, you need to find which feature is the same as the feature in the second data set, because the descriptors, the information we have for these metabolites, they can vary between the studies. And the massSight tries to combine them. So think about the main overview of the massSight, and is a very powerful, useful tools for many metabolomics analysis when you want to try do the meta-analysis, meaning that combining the studies and do the meta-analysis. Here is a cartoon that shows why we need to combine the metabolites and how it works. So let's say you have data set 1 and data set 2. In the metabolomics, usually we don't have the name for the metabolites, we have the descriptors: mass to charge, retention time, and intensity, average intensity across all the samples. Let's say you have n samples in a study 1. Sometimes we have an ID for them, what the name is, but sometimes we don't. We want to see what is each feature, each row, the corresponding row of data set 1 in the data set 2, and we need to combine them. And our goal here is to find, develop a comprehensive data set or joint data set that has n plus n samples, and they're combined. We look at the descriptors, and these descriptors, they might be not exactly the same across two data sets. So massSight has several functions as R package and does several different things. So one of them is auto combine. That is the main function that combines two separate data sets that has been processed separately across different research groups, and you can combine them and do the meta-analysis to find biomarkers with more evidence in terms of research studies. Then the other thing is to do the auto consolidate, which means in this metabolomics LCMS technologies, they produce many breakdowns or adducts that they come from one metabolite. And we want to combine those two as a one metabolite, because that is important to us. So consolidate mean here to see what are the original metabolites in the samples, because the LCMS technology could break them down to a specific fragments or adducts, and this technology or this function help us to get them back and group them based on some of their common properties. And there is one other important function that we have a demo for it, but is not completely implemented, is to scale the data. There are many experimental approaches that they are used to remove the batch effect or drifts in the individual metabolite study. And this function in massSight helps to do that, to use the experimental information that this platform, Metabolomics platform, they use to combine this to scale the data and remove the batch effect in the data. So the algorithm, I'll just go briefly through that. First thing is this algorithm is a machine learning based approach, meaning that try to capture the patterns in the data, and based on those patterns, tries to find what are the corresponding metabolites in data set 2. And the way it works, it tries to find the metabolites that, based on their properties, they are unique in each data set, and they don't overlap, to avoid the confusion in the algorithm. The second part is to model the changes in the two data sets, how features in one data set has been changed in the second data set, and try to model those, because those changes are not linear and they can't follow any pattern. So this approach try to to model the changes, and based on those changes, tries to adjust the data set to try to say what are these values that we have in data set 2 if I adjust for those changes in the data set 1. And then after that, it does an extensive search on searching the feature in data set 1 to find the closest one with some threshold in the second data set. And based on those conditions, generates a combined data set. So in the metabolomics data, this is a key here. We cannot combine two data sets by merging the way we do in R or other computational tools. You need to find the corresponding features, because this MZ, RT, and intensity that we have for each feature, they are not the same for the same metabolites in two different studies because of the batch effects and many changes that could occur during the processing. So we need to model those changes and then adjust the second data set and then find the features in the second data set. So this approach, and I'll mention what are the application for this later. There are many applications for it, but let me emphasize on another challenge with the metabolomics data processing that we are trying to show this mass-Sight approach can be used for that. Let's say you have a study with 10,000 samples or 1,000 samples. The technologies, computational technologies, they are not scalable. They miss the metabolites because at the end for one study, they want to report one metabolite and what is the intensity or the amount of that metabolite we see in different samples. So when you have many samples, then most of those technologies, they fail to identify the metabolites that is consistent across all the samples because many metabolites and many changes over time when they process them. So they are not scalable and the accuracy is very low in those scenarios. That's why the metabolites, when you look at them, those that they have a name are very few. Like you have about maybe 400 if among 20,000 or 900 at most among 20,000 metabolites reported by technology. The technology cannot find the name or even identify exact metabolites. So it makes them separate metabolites. So the scalability and accuracy of those algorithms is low, and the massSight allow us to break down studies to subset or buckets, and then later align them. Basically, you can process each subset of the samples separately. That way, you have a more accurate result for that subset. Then use the massSight to combine them. So this application of massSight is for technology and processing the data, but the main application for it is meta-analysis when you combine two data sets that have been processed separately. So why the massSight is important, especially with the metabolomics? Because metabolomics are expensive in terms of processing them, and also meta-analysis can provide us more piece of information from different studies and find the metabolite as a signature of a disease status. When we find those specific metabolites with the same pattern across different studies. Basically, the several studies, they say the same thing about one metabolite. Sometimes those metabolites, they don't have a name. It's good to see how different studies point to a specific set of metabolites, and then we can map them to each other across studies. So that is a very important function of massSight, to find the corresponding metabolites across different studies when we don't have name for them. Also, this massSight can be used to re-annotate previously profiled metabolites. Let's say you have a study 10 years old or 5 years old. That time we didn't have enough naming or annotation for them. So now we can re-annotate them using the massSight. Basically, we can align an old study to a new study and find what are the corresponding metabolites. And then we can compare the annotation of these metabolites. Sometimes this is a very challenging task to annotate metabolites. We can see how accurate existing annotating methods are. And more important than all this is the meta-analysis. When you add more samples across different studies by combining them, your statistical power can increase because then sample size increase. So we applied this on two different studies. I want to say that there are many studies that they have a similar setup in terms of data sets. Here, this is from my postdoc group that they did work on IHMP, as Brett initially mentioned. There are two different studies that have been processed separately for the same disease, inflammatory bowel disease. PRISM and IHMP are the name of the study, but their setup is very close to each other. So we applied this massSight to show we can combine the metabolites and have a higher power. Because of the time, I'm going to skip some of the downstream analysis. I'm just going to focus on alignment and combining. Here you see these two studies. So this is study 1. You see we have a different cohort and the different health status for the inflammatory bowel disease, Crohn's disease, and colitis. And we have the same thing in the second study. So we combine these two studies, their metabolite part. And when we did that, we were able to map about 2044 metabolites across these two studies. So we were able to map the metabolites across these two studies, and that helped us to do the meta-analysis, because we find among the PRISM had only 2,316, and from these 2,044, we were able to accurately map them to these 87,000 metabolites we had in IHMP. So on the accuracy, the way we check our accuracy, which we put 98% here for it, based on the annotated metabolites. This process is unsupervised, meaning that doesn't take the name of the metabolites in the processing, but we use them at the end. If those that they have a name, they map to each other. And 98% or more of the time, those they map to each other. So massSight is very powerful software to combine metabolites data and do the meta-analysis. And this is a key for many metabolomics studies, because if you find a metabolite that is important, has a role in the health conditions, by doing the meta-analysis and combining the different metabolites profiles and doing the meta-analysis, we are able to find the higher or more evidence for those metabolites if they are consistent across different studies, to report those metabolites with a higher confidence. So that was the second package or second approach in metabolomics. These are novel techniques that allows combining and doing those applications that I mentioned. Now I'm going to move to another techniques, and that's going to be the last one. I'll be brief this. The concept is very simple, but very important. Also a little bit complex in terms of computation, and we try to make a user-friendly software for all of these tools. So the omics dynamics, this is a key in many omics, not only metabolites, all the omics. When you have a longitudinal study, and the question we want to ask here is, if we process the metabolites in the longitudinal data set, and the question we want to ask is two things. Number one is how the measurements, the things that we have measured, the microbes, for example, or metabolites that we have measured over time, how they change. We need to find the functions, temporal dynamics or functions, that they explain the changes over time using the data sets we have measured. And then we find those functions. We might go with 5 specific functions that they explain the changes of our features over time. Then after that, the second question the first question is to find those functions of changes or temporal dynamics. The second question is to find the associations between the omics features with those functions. We want to say those functions that we described and we characterize in our data set, how those they change, how the omics features like metabolites are related to those changes or functions. Here, we look at the ocean microbiome, and we find a lot of good evidence that microbes, they change in different time points of the collection. We had a sample for the studies, and I just want to show how the function and the method in general works. So your data sets, this is a work that Alan Ross, a PhD student in the group, with Ariza Tairion, a postdoc in the group. They are working together to develop and test this software for many, and they are applying it on many studies. And the way it works is, let's say you have observational data, then you want to see and characterize the function of changes. These kernels that you see 1, 2, 3, 4, they explain the function. The easiest one is kernel 3, that is a linear change. And each point, each color here represents one subject. So we see among all the subjects that we have measured their changes, that we see a change that is linear over time. But the changes are not always simple as that. In the studies, we want to find all the functions, like the kernel 1, kernel 2, kernel 4, and 3 as well. And the data set usually comes like this. You have the IDs they could be the individuals. Then you have a time, which time point we have collected this sample for each individual. If they had a treatment, you see there are many of those. And Y1 and Y2 are the outcomes. Those could be the metabolites, and they are high dimensional. So we just use the Y1 and Y2 to explain that we have many outcomes over time. And the question is to see what are the functions that explain the changes in Y1 and Y2. For example, in Y1, we see the change is related to the ID, which the ID here is the subject. So you see there is some complexity here in terms of how we can find the dynamic of changes and how they are explained. This part is very simple and straightforward in terms of explaining the concept we are trying to, or the challenge we want to tackle in this technique. So again, I want to emphasize in Y1, what you see here, Y1, for Y1 is one of the metabolites, let's assume, and we see the metabolite changes are consistent, and those changes are about the object, the IDs. The people, they have a different metabolite. That metabolite doesn't change over time for each individual, but is different in each individual. That's one dynamic that we are talking about. And you see the data set look like this. We have IDs, if we have a treatment as well, if we have a time, and Y1, Y2, the software is generic, meaning that if you have the table, regardless of the title of this column, if time is there or not, tries to characterize the changes, and if you have time, you can interpret the result based on the time and find the associations. So again, this approach does two tasks. Number one, find the dynamic of changes. Second is tries to explain how our omics features are related to those changes. So maybe you ask, and I have a few more slides, I'll be done quickly. So if you look at this cartoon that we try to show a dynamics, when we say dynamics, what they mean. So for example, the first one in the left is a linear change. Linear change is a dynamic of, which is linear, is very easy to explain. Periodic changes, it look like this, but the combination also can happen, which you see a linear pattern, and over this linear, you see the periodic change as well. Sometimes the change is not a plus, is a time, and that's how that's look like. You see in this cartoon, we try to show the different dynamics that can be hidden under or observational data. And we try to capture these dynamics, these changes, and then we want to see what they are, because you can have an infinity number of functions. We try to find those that we have evidence in the data for them, and that's we call it kernel search. And after that, we want to see what are the omics feature here for us. Metabolites follow these patterns and maybe have some meaning for us. For example, in soil microbiome or metabolites, seasonality is the function sometimes. And we want to see if the microbes or metabolites, they change in different seasons. Those could be the application of this. So I'm just going to show you an example here for a different study, breast milk. In this study, we try to characterize the breast milk microbiome and infant health. We collected a stool sample from infant. We collected the breast milk from maternal, the biological moms, and donors. And we had many hypotheses and questions. And because we had a longitudinal aspect here, we tried to ask if these metabolites from the infant, they are associated with some of the covariants over time, because we had a longitudinal. Some of the hypotheses were very clear, and we had evidence for them. For example, for this particular microbiome species, that was several of the main hypotheses. The longitudinal part was the secondary hypothesis. But here we showed some evidence for the microbiome, if you are interested. We showed some microbes. We hypothesized that they transfer from the breast milk because they had a similar genome when we look at the genome of a species in the breast milk versus the infant and stool samples. And we found their genomes are very similar. We hypothesized that this microbe, they transmit from breast milk to the infant. But that was a major hypothesis that we test, and we find evidence for it for the specific microbes. But the secondary hypothesis, which is the outcome of is the second the hypothesis in the study is to find the dynamics, number one, and then see how the metabolites are related to these dynamics. So what we see here, this is the outcome of the waveome. Waveome is the name of the software that does find the kernels or the dynamics and their association with the metabolites that we are measuring in our study. So here in the row names, or here you see them, the row names are the dynamics that they are explained by the covariates, the measurements we have for the samples. For example, here it says the breast milk, how they have been collected from the donor or from the maternal. That's a one big function in our study, that the measurement of the metabolites is related if they come from the breast, directly from the breast milk, or they come from the donor. The other thing is related to the individual IDs from people, who the individuals are, and those dynamics or changes are related to those. So that's the one thing. And the last one here, we see a change that is related to the age at the breast milk sample. So we find the dynamics actually in this study that they are not related to time. So the time was not exactly the main thing, except for the age at the breast milk sample, which reflects the time of sampling. And we see some dynamics, squared exponential was the dynamic of this change. And we see here in the y-axis, or the columns, are the name of metabolites that are related to this dynamic of changes. This heat map basically shows the dynamics of changes and how the metabolites are related to those changes. Yeah. In summary, what we try to here in summary, what we try to do is we show that how the metabolites are increasing, the use of them, and why they are important to do this, apply these computational techniques on them. The second thing is we show that Tweedieverse is a computational techniques that can help us to do appropriately measure the differential analysis. We can perform differential analysis more appropriately for the omics data using the Tweedieverse, because look at the property of each feature individually. massSight also help us to combine the metabolite data sets when they are occurred separately. They are coming from different data sets. And the lastly is we discussed the waveome. Waveome is a computational techniques that allow us to ask some specific questions when we have a longitudinal data from metabolomics or other omics. We try to find the patterns of change and see how our omics features are related to those patterns. So most of these tools that we have developed, I skip one part, a big part in our research, which is all of these questions and these tools are motivated with the biological questions. And these two, they go together. So developing software and having a hypothesis, biological hypothesis to address them. And we have applied these techniques on many different studies that I skip because of the time in this study. So with that, I want to pause here, and these studies that I showed, they are from collaboration I have with the Miriam Bornhorst from the MEKi, and COVID-19 was with Keith. And these are the students and trainees in my lab that they have helped with these analyses. We have many other collaborations, like the single cell with the bread that mentioned that we use the Tweedieverse on them, and we have a lot of good evidence that these softwares and these packages work very well. With that, I'm going to pause, and if we have time, I'll be happy to take questions.
Thank you very much, Ali. That was wonderful. I hope that everyone was able to grasp the significance and functionality of these tools, because I think you presented that very clearly. But I hope that people also can take away how accessible you're making these tools for researchers. If anyone has any questions, you're welcome to unmute yourself or put them in the chat, while we give people a chance to formulate some of these ideas. I'll fill this time a little bit. So Ali, with a lot of these computational tools becoming built into R so easily, and that being more accessible for researchers, more and more people are just following standard pipelines and becomes really daunting to try to figure out sometimes which of the available tools are the right one to use. So you did a great job of showing with Tweedieverse that genes within the same data set can have really dramatically different distributions. And it makes a strong case for why you'd use Tweedieverse. Are there any conditions where Tweedieverse wouldn't be ideal, or where standard pipelines are still, I'll say, acceptable with this distribution?
Absolutely. So that's a great question, Brett. So there are many tools like general linear model-based approaches. If we know that the data sets follow a specific distribution, then many other software, they can do a great job there and in a less time. Because the Tweedieverse, one of its challenges is that for each feature, and that's why if you have used Tweedieverse versus the other tools, you see Tweedieverse takes longer time, because for each feature tries to characterize the distribution of that features. But if you know the feature, they are following the same distribution, then general linear models like Maslin-2 that we also were part of it, or dissect-2, they can do, there's a name of other software, they can do a good job on that, and they have a more, they can do a similar performance.
Excellent. Are there any questions from those in the audience? All right, then I'll ask another question, because I have another question. I have multiple questions, but I'll ask another question. So with Tweedieverse analysis, you mentioned that one of the big changes that were discovered were some enzymes that are used for generating specific lipid species. And this really got me thinking about how so many new lines of investigation are incorporating a multi-omic approach. So with Tweedieverse, is this something that can be used also for things like lipidomic analysis instead of transcriptomics? And if not, what would it take to be able to use something like this package to apply to a different omics, or how well do some of these integrate?
Absolutely. So, Brett, this Tweedieverse, we have tested for different omics. Like you are aware that we did work together the single cell data. We have applied on the metabolomics and microbiome data. So Tweedieverse is generic, meaning that you can apply it on any omics data and is not specific to any omics, because the character it has tries to characterize the feature of the distribution of each feature, regardless of where it comes from, is metabolomics or microbiome. And also they had the other big feature, which is the Tweedieverse models that we use in the background to do the differential analysis. I forgot. They don't need to normalization. So you don't need to normalize the data. And the normalization is a big task in omics regardless. So it depends on which technology you use, is single cell or metabolites or microbiome. Normalization, it could happen in different ways. Tweedieverse doesn't need that. So you don't need to normalize. So you can just feed the raw counts or any positive values to it.
Fantastic.
And I just want to add this to the group, that these softwares are available online, and we do support, if you use them and you find them useful, we can do a specific meeting with the people. If the researchers, they have questions, we'll be very happy to sit with you and help you to use them and run them if you have a question for them.
That's fantastic. Thank you, Ali. Hearing no questions from the audience, seeing no hands raised or anything in the chat. I'd like to thank you, Ali, for this wonderful presentation on behalf of the Office of Integrative Medicine and the Department of Biochemistry and Molecular Medicine. We're very grateful for this. And a recording will be available to individuals who were in attendance and for those who registered. So thank you very much. And I hope that everyone is able to continue attending the cross-disciplinary seminar series on metabolites as we continue this throughout the year.
Absolutely. And thank you all for attending.
Great.
Bye.
Bye, everyone.